Dr. Claire O'Callaghan, who's our next speaker. She's a clinical research fellow with a focus on neuropsychiatric symptoms in neurodegenerative conditions. And her research takes a transdiagnostic approach across different conditions, including many neurodegenerative disorders. I'm going to stop because my dogs are going to start barking. So, Claire, I'm going to hand over to you. You're going to speak to us about translating neurodrenic. Oh God, I can't even say
Noradrenergic. Don't worry.
neurodegenerative diseases. So I'll hand over to you.
Thanks so much. Cool. Let me just share my screen. Okie dokie. Okay, I'm assuming everyone can see my screen and can hear me. Great.
Yeah, so we can see everything.
Right. So I was really happy when I got asked to when I got invited to speak. So thank you for the invitation. I was happy because the overarching theme of translation is something that I love to think about. And I guess everybody has kind of gleaned by now in the symposium, there are different types of we talk about different types of translation as it applies to our research. And the particular type of translation that I want to talk to you about today is the idea of cross-species translation, with the notion that when we can think really carefully about ideas around cross-species homologies, it puts us in a really amazing position to get new insights about the brain basis of behaviour, and crucially, for the work we do at the Brain and Mind Centre, new insights into understanding cognitive and psychiatric symptoms in different types of diseases. So cross-species comparisons can happen at a lot of different levels. It can happen at the level of comparing structures, so thinking about, cytoarchitecture or receptor architecture. We can think about connectivity across species, so whether this be sort of hardwired axons or the more sort of dynamic measures of connectivity. And we can also think across species at a functional level. So thinking about the sort of fundamental processes that a region or circuitry gives rise to, and how that then sort of shapes behaviour and psychological processes. With the idea being that when we can sort of weave together these ideas of analysis, we can gain really important insights so we can translate things from human into animal, and also back translate from human back to animals. Now, when we think about, I guess, sort of cognitive and psychiatric symptoms, we're probably particularly interested in sort of nailing down animal models where we think that the function or the behavior at hand is, analogous to a function or a behavior that we're measuring in people. And with the idea being that we can have this behavior that transcends species, and that it might be then able to be mapped onto similar structures and connectivity, which then, opens up possibilities for us to, say, test out new drugs and new treatments, for example. Now, one really cool tool that allows us to do this is the notion of cross-species tasks. So that's the idea of a sort of task that measures a behavior that we can deploy across species. So whether it be rodents, monkeys, or humans, for example. Now, one example of this kind of task is the stop signal task. So you can see there on the left side of the screen, a rat is diligently doing the stop signal task. And of course, the human version is much more boring. It essentially involves asking people to stare at, a long string of arrows, and they have to make a response indicating whether the arrow is left or right. And every now and then, a tone will come on that tells them that they need to cancel their response. So they need to stop responding. So essentially, this task gets at the idea of inhibiting a response that's no longer appropriate to a given context. Now, this might be operationalized in a reasonably sort of simplistic way in this task so that a mouse and a human can do it, but it's actually quite a fundamental behavior. This ability to stop an inappropriate response and, to sort of reconfigure the action that you're doing is actually something that is, a really fundamental part of our adaptive and flexible behavior out in the world. And it's an ability that goes awry in a whole host of different diseases, particularly some of the diseases and disorders we study at the Brain and Mind Centre, ranging from psychiatric all the way to sort of neurodegenerative diseases. So the kind of important thing about this task is whether we're giving it to a rodent or a human, we're able to distill the same metric from it. So we're able to quantify just how fast or how efficiently a rat or a human is able to stop their response. So this is called the stop signal reaction time, and it's a metric that we're able to use across species to indicate how well that response is inhibited or controlled. So through loads of beautiful cross-species work using this task, we're pretty convinced that it does actually map onto the same processes in humans and in rodents. And these are the processes of response inhibition and impulse control. Now, what beautiful cross-species work with this task has also offered us is the idea that, in fact, this task seems to tap into similar connectivity or circuitry across species, namely a sort of corticobasal ganglia circuitry that is, particularly involved in inhibiting responses in this way. And even more beautifully, from very careful cross-species work, we note that there's a homology in terms of the neurotransmitter systems that seem to come on board when this type of behavior is required. And in particular, the noradrenergic system sort of persuasively modulates this ability to withhold or cancel a response. So you can already see by using this kind of task, we can weave together these three levels of analysis where we're able to sort of go all the way from a behavior, and particularly a behavior of interest in many different diseases, all the way through sort of the structure or the brain areas involved to neuromodulation from the noradrenergic system. And that allows us to think about, well, what diseases, have impairments in one or many of these sort of layers of analysis. And relevant to the work that I do and many others at the Brain and Mind Centre, we know that neurodegenerative diseases of aging have impairments across, many of these diseases have impairments across all these three levels. And I'm going to focus in particular on Parkinson's disease, which many of you would know, of course, as ostensibly a motor disorder, but equally troubling and pervasive as the motor symptoms are the cognitive and psychiatric changes that happen in Parkinson's disease. Now, with good reason, and for a long time, the focus in Parkinson's has been on the dopaminergic system. So we have loads of good treatments for restoring dopamine function in Parkinson's disease. However, the Locus-noradrenergic system is affected really early on in Parkinson's and really quite profoundly affected. And as I mentioned before, the noradrenaline system, which is implicated in this type of response control, the locus coeruleus nucleus in the tiny nucleus in the brainstem is actually the brain's main source of noradrenaline. So impairments or early changes in the locus coeruleus, it gives you this sort of widespread impairment of this system. Now, we're actually able to image the noradrenergic locus coeruleus in Parkinson's by virtue of a special property in this nucleus, neuromelanin. And by using sort of high field MRI, we're able to get a measure of the integrity of the locus coeruleus. So bringing all these together, we were able to sort of take a translational, or inspired by this translational approach, to see if we could use noradrenergic drugs to improve cognitive function in Parkinson's disease, and sort of weaving them together with the stop signal task based on the sort of animal work that I've shown you already. We had a second question as well. Not only did we want to see if we could use this task to measure the effects of a noradrenergic drug, we also wanted to sort of try and pinpoint who this drug might work best for. And we had the notion that integrity of the locus coeruleus noradrenergic system might give us a clue to sort of help guide us who might benefit most from this sort of therapy. So we designed a study where we scanned people and measured the integrity of their locus coeruleus, and then we got them in on another day and gave them a dose of a noradrenergic drug, atomoxetine, which boosts noradrenaline levels in the brain. And we tested them on the stop signal task, either on the drug or on placebo. I'll just skip that little bit because that's referring to the sort of modelling that we did to give us estimate the stop signal reaction time. But what we saw in terms of behaviour in our Parkinson's patients, all of the basic levels of performance were unaffected by the drug. However, when we measured the stop signal reaction time task, we did see that patients were less able to withhold their response. They had sort of problems with their response inhibition. But you'll note that actually both the placebo and the drug group were worse than controls, and there wasn't really a difference between them. And this comes then to our sort of secondary hypothesis, where we thought, the integrity of the locus coeruleus-noradrenergic system might actually sort of explain who would improve or not on the drug. And indeed it did.
You've got 1 minute left.
Yep, indeed it did. So we found that there was an interaction where, to sort of unpack it in more detail, people who had a more impaired locus coeruleus, when we gave them the drug, they improved much more than the people on placebo. So this was able to give us a really important clue that intervening with a noradrenergic drug in Parkinson's, there may be a particular sort of sweet spot where impairment of the locus coeruleus can determine whether you'll actually benefit from the drug itself. And we can already see that not only does that have implications for guiding precision and individualized medicine, it can help us shape clinical trial selection in terms of selecting people who may be more likely to benefit from the treatment. And I'd just like to thank the sort of huge team effort that was involved in that study as well. Thank you.
Thank you, Claire. You covered a lot of ground in that 10 minutes.
Probably too much.
Congratulations on that presentation. It was a very elegant description, I think, of linking functional behaviour with circuitry with the neuromodulation. I thought you gave a brilliant description of that. So thank you very much. We might move on just on the interest of time, just because we need to get to our last speaker. So thank you very much. Great presentation.